I'm a biologist, and I got involved into bees when I was asked to judge a project on artificial selection for resistant bees that were resistant against Varroa. And that's a bit where my story in bees begins, and that's why I also have this first lecture on that. And you'll see that tomorrow we have a very different talk, but then on Saturday I come back to this subject, and then we go really deep into the genetics. Today we stay a little bit at the surface. So first, where does the Varroa come from? It comes from another species of bee. So it co-evolved with another species of bee, and that's the eastern honeybee. When parasites and host evolve together, they get adapted to each other. And so on the eastern honeybee, there is no problem. The host and the parasite can coexist stably, and the mites are always at a low density. And we'll see why in a while. But the European honeybee that was invaded by this Varroa has problems. The parasite, when it's present in a European bee colony, it eventually will kill the colony. And the mites, they grow exponentially. And so we still treat bees against the Varroa mite. And still there is a lot of mortality. Now let's see what happened. This is the distribution map of the eastern honeybee. And the interesting thing is that it crosses into Russia at the very northern edge of its distributional area. And that's a pity, because if it would not have occurred in Russia, we would not have the Varroa problem. Because what happened was that in the beginning of the 20th century, a lot of Russians moved to the east because there was a lot of economic possibility. They had discovered silver in the ground, the sea was full of fish, and there were extensive forests that could be harvested. So people moved, and they had done that before, but then it was only possible on horseback, and it takes a long time to cross Russia on horseback. But by 1904, the Trans-Siberia Express existed, and so people came by train to colonize that region. And the other advantage of a train is that you can take your bees with you. So they brought the Western European bee to the region of the Eastern European bee, and because the Eastern bee had turned out not to be very pleasant for beekeeping. And then the problem occurred that the mites could move from one species to the other. And then in the 1950s, people started to move back to Moscow, and they took their bees with them because they thought that these bees were excellent. They produced lots of honey. But when they arrived in Moscow, they turned out not to produce a lot of honey. The property of producing a lot of honey was a property of the extensive lime forests in the east. And those lime forests are not present in Moscow. So they brought the bees for the wrong reason, but they brought also the Varroa mite, which then spread all over Europe and over most of the world, and now is also present in Australia even. There are important differences between the two species of bees. One is that the eastern honeybee has a much shorter developmental time, which means less reproductive opportunity for the mites. And in the eastern bee, there is no reproduction in the worker brood. And in the western bee, most of the mites are produced in the worker brood, but also, of course, in the young drones. The eastern bee all the colonies have defensive behavior, and so they remove mites that sit on adult bees by grooming themselves, but also by inviting sister bees to do the grooming if the mite sits on a place where they cannot reach it. And they have also hygienic behavior. They remove cells with developing mites and they kill the young bee. Now here you have a family of Varroa mites. This is a male, and these are all young females. The male is there to fertilize its sisters, and then he dies, and the sisters move on to cells to make a new cycle of reproduction, and the mother also moves on to a new cell for a new cycle. So later we'll talk about the reproduction per reproductive cycle, and then that is what I mean. One reproduction by a female mite. Now, and here is the problem that I got stuck with for a long time. In Europe and in the USA and in Canada, beekeeping is not possible without a form of anti-Varroa treatment. And you have all these colony losses. But in Africa and in South America, Varroa mites are not a problem. They've colonized those places, and obviously natural selection has taken care of it. And then the question is, why does natural selection not take care of the same problem here and in the USA? You have to understand that bees are quite exceptional animals, a little bit comparable with salmons and with oysters and mussels. They exist in the wild, and at the same time they are bred and cultivated, and so they're not a wild species or a domestic species, but they are both at the same time, and that's very important. Also, we should realize that honeybees are a native European species, and so that for a long time the wild colonies were subject to natural selection, but also the colony and the beekeepers, maybe they selected bees for their own purposes, but those bees would interbreed with the wild population, and swarms would return to the forest. So natural selection was also part of the practice in apiculture. But now we have the problem that after 40 years, we have no population-wide resistance. First I thought, well, that's very clear, very simple. Beekeepers, all the things that beekeepers do work against natural selection. So there is chemical treatment, but there is also requeening. But other apicultural practices also are against natural selection. A lot of things that beekeepers do is preventing the bees to reproduce. Beekeepers don't like swarming, and swarming is the way that bees reproduce. Other people also realize that it's very difficult to study natural selection in honeybees that are kept in apiculture. And so a number of researchers already, what started 30 years ago, they wanted to do experiments on natural selection. And you can do that by having bees that you don't treat and that you leave over to what the mites are doing. And so there are a number of long-term experiments without treatment, and I want to look at those. By comparing what happens, we have to look at this value, and that is the number of fertilized daughters that is produced by one breeding cycle. And Martin used this value WR to calculate what would happen with a Varroa population inside the bee colony. And he could show that when the value is above 0.7, then the Varroa population starts to grow exponentially and eventually is killing the colony. And only when the value is below 0.7, then the Varroa mite can be controlled by the bees. And here you see how that works. So in Apis cerana, the eastern honeybee, there is no reproduction in the workers. And so the WR is 0. In the Africanized honeybee, it turned out that the WR was exactly 0.7, but not in the European honeybee. It was either 0.9 or in this experiment, but I'll show you also figures that are much higher. So here you have a summary of all the experiments that people have done. Most of them started by the observation that bees, for some reason, had survived an attack by Varroa mites. And then these bees were taken to an experimental, into an experimental design, and they were studied and they were kept in such a way in the hope that natural selection would advance and that these bees would become resistant. And so we have an experiment, a large experiment in Sweden. On Saturday morning, I will talk in more detail about that experiment because there's some very interesting things inside there. And then in Norway, they discovered a population that persisted without treatment. And the same happened in two places in France, one in western France and one in the southeast, in Avignon. And if you look at these WR values, you see that these bees cannot be resistant. The mites grow, the populations grow, and so the colonies must die. So it's rather sad that all those experiments, in fact, if you start to calculate what happens, is that they are better than the bees that were not in the experiment, but they are far away from being good enough to be resistant. So that was a rather sad conclusion. Now, what can bees do to be resistant? They have this Varroa-specific hygiene. That is, you have bees that are able to recognize from a closed worker cell if there is a mite inside that is reproducing. And then they open the cell, and then other bees come and they remove the bee pupa with the developing mites, and they die. And the mother tries to reach another cell for a new cycle, but maybe she's grabbed by a bee and killed, and then she falls down. Then you have this allo and auto grooming, which is very important for bees in the winter. When there is no brood, then the only place where the mites can survive the winter is on top of an adult bee. And so it's sucking hemolymph from such a bee, and it means that that bee has a smaller probability of surviving until spring. And so that's not a good thing. And then very important, the Varroa mite is a vector of a number of viruses. Deformed wing virus may be the most important one. And those in the end kill the bee colony. So being resistant against the virus would already help a lot. These are all heritable traits, so you could select on them. Now, swarming is also partly heritable, but it also depends on the size of the hive and the health of the bees and the amount of stock they have. whether they swarm or not. But swarming means that reproduction by the bees is stopped for a while. So for the queen that leaves, she first has to go to a new house, which has to be built up. And so she is for 3 weeks out of business. But the young queen that leaves behind in the old nest, also she must go on a mating flight, and it takes a few weeks before she starts to reproduce again. So there's a gap in the reproduction, and during that gap the mites also cannot reproduce. Now here you see in a picture this behavior of Varroa specific hygiene, and at the left you see a cycle where the bees just suffer from the Varroa mite that reproduces, and when the young bee comes out, there's also a whole family of mites coming out. Now the only experiment, I showed you that list of 4 different studies. 3 of them, there are all sorts of problems with the experimental design. But there is one outstanding experiment, and that was the one done by Ingmar Fries in Gotland. Now the interesting thing is that in his experiment, none of the bees had alleles for this behavior, this hygienic behavior. None of the bees in the whole experiment had this character. On Saturday, I will try to find an explanation why that was so, and it's a serious business. But for now, we have to remember that this was so. And the colonies in his experiment that survived, they were small and they swarmed a lot. And we understand that helps a little bit against Varroa. And they were highly inbred, and you all know that inbred bees are not good. So we are not surprised that our honey production was quite low. There was one thing which was interesting, is that if you took a frame and you opened the cells to find the cells that contained Varroa, there were a lot of cells that contained a mother Varroa, but that mother had not reproduced. No eggs, no young. And that is interesting, because if they don't reproduce, they're not so harmful. And the idea in this case is that the lack of reproduction in the mites is caused by the fact that normally the developing bee produces hormones that signal when it is going to pupate, and that is also a signal for the mother mite to start laying, and then the whole cyclus is synchronized with the development of the bee. And the idea is that these bees, for some reason, produced less of that signal, so that the mite missed the signal and did not reproduce, and sat there waiting for something that didn't come. When all those experiments were done, the European Union funded a large experiment where bees were moved between countries in Europe, and they were not treated against Varroa, and then it was studied which bees did better. And the question they tried to answer was whether there is such a thing as local adaptation. So the hypothesis was that bees that originated from the same place where these hives were standing would do better than bees that came from other countries of Europe. And so they had swapped all these hives, and then they looked at what happened. And it's one of the most depressing publications I've ever read, because you see that after 2 years, all those colonies are dead. And what is also sad is that here we have the Avignon, and that was one of those strains of which Leconte published that it was resistant against Varroa. But when in this experiment, you see that it's absolutely not distinguishable from all the other strains that were supposed to be vulnerable for Varroa. So it's not a very convincing matter, those experiments. And then you understand why, because then he writes in 2070 that he was keeping those, between quotes, resistant strains, but that they were allowed to mate freely with the bees that lived in the surroundings, and that he was breeding a lot of queens so that the bees could not go extinct. He would replace the queens. But if you do that, of course, you are opposing natural selection. So if there was any resistance in those bees at all, after a few generations, those genes have been diluted over the environment. The mortality rate of the surviving colonies is not different from Varroa-sensitive ones, and the beekeeping practices in those experiments have interfered with natural selection. All those experiments we were talking about is colony-level selection. And so we must conclude at this point that colony-level selection has not resulted so far in useful resistance. And then we have the next question, why is that? Why doesn't it work? Now, in South Africa, it did work, and in South America, it also worked. And then there is this other large experiment. Because the Varroa mite has been introduced from the very east of Russia, a region that is called Primorsky, Americans thought that those bees that live there had the longest exposure against Varroa, so that you might expect that in eastern Russia some resistance has developed. And so they went there, selected queens, they pre-selected queens that they brought to the US, then selected further, and I'll tell you the results of that study. And in the end, they were resistant, but you see that it has been quite a struggle. And then we know from Thomas Seeley that the bees in Arnold forests, which are feral bees, the bees living in the wild, so they are not treated and they are exposed to natural selection. And he could show that first when the Varroa mite arrived, these local bees collapsed, but some survived and some new bees came in, and in the end of the story was that 10 years later they were resistant, and they showed the behaviors that I mentioned that are used by bees to defend themselves against Varroa, the hygienic behavior and this allo and auto-grooming behavior. Now let's go to South Africa. So in the south of Africa you have two subspecies. The very south you have the Cape honeybee. Then there is a band where you have the subspecies Scutellata. And both those subspecies became very quickly resistant. In 5 years' time, the Varroa mite spread from the harbor where it was found first over all of South Africa. And it was very dramatic. During peak infestations, they had hives with 30,000 and even 50,000 Varroa mites in one hive. And on average, the number of mites was 10,000 to 70,000 mites per hive. So you can see that these bees were absolutely not resistant and did not show these behaviors when the mite arrived. But the good thing was, very different from what happens in Europe, not all the colonies died. We don't know for sure, but it looks very much like those bees were resistant against the viruses. And so they could tolerate very high densities of mites and survive. And for that reason, natural selection could do its work. And 5 years later, the problem was gone, and I wrote to Mike Alsop about it, and he said, now, he said, it's no more than an arbitrary presence. There are a few mites in each hive, but we don't care. They are not harmful anymore. And now these bees, they have this hygienic behavior, and they have this grooming behavior, and in that way they're very resistant against the Varroa mite. The problem is over. Now the good thing was that when the mite arrived in South Africa, Mike Alsop started on a master project to study the spread of the Varroa. So in South Africa, the problem is very well documented. We know exactly what happened. Unfortunately, we do not know what happened in South America. We know now that the bees are largely resistant against Varroa, but there is not a very good documentation. But we know that the mites arrived in '79. There was no large-scale colony mortality, and also, something I was really surprised to read, that in the tropical forests in Brazil, the most common pollinator is the honeybee. It is an introduced species, but it's everywhere in the forest. And so there are many, many, many wild colonies of honeybees in Brazil, allowing natural selection to work. If you look at what the incidence of hygienic behavior and grooming behavior, you see that that's very high in these bees. And in fact, every time you find bees that are well resistant against Varroa, they have these two behaviors. So they're really instrumental in the resistance of the mites, of the bees. I was for a project in Panama, and so I went to see some Indian beekeepers to ask about their experience. They don't treat against Varroa anymore. And also they had scared me by saying, if you go with the Indians and their bees, you will be in danger because these Africanized bees are very aggressive, and you might get a lot of stings. But I was surprised to see that these bees are very gentle, and during the inspection of their hive, they sit on the heads of these people that are doing the work. They were not at all aggressive. And now the story about the Primorsky bees. So these honeybees, they arrived in Primorsky in the early 1900s. The first, at least in 1904, we know, because the first train arrived in 1904. In the 19th, the Americans went to that area to look at the bees, that the western honeybees that were still present in eastern Russia, and they looked at whether they were resistant to Varroa or not, and they found that they showed varying degrees of the two behaviors that I mentioned. And so they collected pre-selected queens, 362, and they were brought to North America during 5 years. They selected artificially on those bees to improve the resistance. The result was rather disappointing, because already in Russia they had this enormous variation. But this is after 8 years of selection in America, which is after the pre-selection they did in Russia. And you see, some of those are very resistant, and you have a very low population growth of the mites. But there is an enormous variation, and there are a lot of them that are still absolutely not resistant against Varroa. So it puzzled me. I thought those results, after all that work, were rather disappointing. And then the USDA, who did the selection work, said, well, okay, we have worked on this program so long, and we are researchers, And it's not our task to make bees for the beekeepers. So we give those bees away. And there was an association of Primorsky beekeepers that then made, in isolated forests, which you can do in the USA, they made mating stations and they kept those bees there and they let them interbreed freely, and in that way natural selection could again have a go on the resistance. And then you see, and now we are 18 years after the bees were imported, that Primorsky bees have become very resistant in comparison with the controls. So it works, and those bees also have the two behaviors. So you see that the resistance was only achieved when these lines of bees were selected for in groups in isolated mating yards. And so there seems to be something with the population size that makes that natural selection works or not. Now the idea is that the population mating structure of bees, the bees have a very interesting mating structure. Ecologists call it panmictic, and panmictic means well mixed. So you have a well mixed population, it's a large population, and bees from a large region, they can mate. And that is because the bees have places where the drones gather, which are visited by the queens, and that makes it possible for a queen to mate with a drone that lives 15 kilometers further. And because not all drones from a colony visit the same mating site, you could think of this as even further extended. And to explain that a little bit more, Tom Seeley has calculated something that evolutionary biologists use. It's called the effective population size. Because you can imagine that now all the bees in England could mate with each other in principle. But there is, of course, there is isolation by distance. So most drones that mate with a queen, they come from an area of about, with a diameter of about 7 kilometers. And only 10% of the drones mate with queens that are further away. So that means that you have a sort isolation by distance, and then you could say, but how should I then define the size of the population that is used by the bees? What do the bees experience as their population size? And there are formula to calculate that, and the conclusion of Tom Seeley was that the Arnold forest bees, they have an effective population size of more than 600 colonies. And so more than 600 colonies is a size where you can have evolution of resistance by natural selection. But now, and that's very important to know, imagine that you would like to select your bees for resistance against Varroa. I always say, beekeeping, you don't do that alone. Because if you're the only one who is selecting, and around you there are all beekeepers that replace their queens every year and keep expensive breed bees, then you may succeed in having some selection, but the genes, the alleles for resistance, and dilute over the population that does not have that. And that is also because of that large mating population. So you can only succeed in selection when you are with a group of people and all the bees in an area are exposed to natural selection. What you want is that the frequency of resistance allele increases in a population. And that could happen in South Africa and in South America and in Arnold Forest, because there were many, many colonies in one population, all exposed to natural selection. Now, natural selection can also work in closed populations, and that's interesting. So imagine that you are the only one who has been selecting resistant bees, and you have done a very good job on your own. You've done it by artificial selection. But now you let your bees mate with the drones that surround your hive or your hives. And what you get then is that if the average frequency of a resistance allele in the population is 20%, then your bees also go down with each generation until you hit the bottom line of 20%. You can also do stronger selection, but in the end, there's a point where it stops, where the dilution is equal to what you gain by selection in a generation, and then you can't go any further. And in the African bees, you can imagine that half of the bees survived already, that there is dilution, but it does not go below the 50%. But if you then select, then you might lift the whole thing and everybody may become resistant. And now we have this example from the Netherlands that I will give you. We have a chain of islands in the north of the country, and two of those islands, they have still good populations of the black bee. And those bees have been treated until about 2005. And then the beekeepers, they stopped, or at least a large number of beekeepers on site stopped treating for Varroa, allowing natural selection. And we know exactly how many colonies there are on the island, and then you can see that if you select on a number of colonies, large enough number of colonies, because it's a closed population, then in your selection line, the resistance goes up, but in the general population, it goes also up, because the bees cannot go outside the island. And so we have these two examples where now have black bees on the Isle of Texel and the Isle of Terschelling, and they are both resistant against Varroa. They have a high incidence of hygienic behavior. There's no need to treat them anymore. And interestingly, you can have also effective natural selection in a large open population. And that's already the example of Tom Seeley's bees in Arnold Forest, because there we had an effective population of over 600 colonies, and they became resistant. But in the north of France, where I keep bees myself, is a region of poor farmers, and they used to produce cider. And so they had orchards, and they all had bees, but they were just the wild bees. They would collect swarms, and their bees would swarm and fly to the forest. And it's a landscape with hedges, and in these hedges, because they have been cut during hundreds of years, there are lots of hollow trees, and there are lots of wild bees living in those hollows. And so there also you have a very large population which is exposed to natural selection and not treated against Varroa. And it turns out that these bees are resistant against Varroa. Now we talked briefly about swarming, and here is the experiment of Tom Seeley and his co-workers that showed that if you look at colonies that were in the same place, in the same bee farm, when they did not swarm, you see that the Varroa mite reaches quite high peaks, but when they swarmed, you see that the Varroa mite cannot reach very high densities. And the difference is due to the swarming produces a break in the reproduction of the bees, and therefore also in the reproduction of the mites. And then he had, interestingly, he had two situations. One situation where groups of hives stood close together, and another where the hives were dispersed. And then you see that if hives are close together, the mites move between the different colonies, and that's also better for the mites than for the bees. Derek Mitchell pointed out to me an old Dutch experiment which I had read at the time, but I couldn't think how it would be important at that moment. The experiment shows that mites reproduce very badly at a high relative humidity. The difference is really dramatic. Derek also showed that in natural wild bees that live in hollow trees, the air humidity can be much higher than in our beehives. So our beehives are good for Varroa reproduction and not good for the bees. And if you manage to build a beehive in which the air humidity is much higher, then also you make that the mites cannot reproduce so well. Well, if the mites have a lower reproduction, the bee colonies will survive longer, and for that reason, natural selection has more time to work on those colonies. So I think that this difference has also contributed to the fact that the bees in Arnold Forest have become resistant against the Varroa mite. And here is a summary. Wild bees, they can survive Varroa infection better than bees kept by beekeepers because their colonies are isolated by distance. In very good habitats, you can have 3 wild colonies per square kilometer, but most often it's down to 1 or less than 1 colony per square kilometer. So it means that there is an enormous distance between colonies. Those wild colonies, they swarm more often than the bees kept by beekeepers because beekeepers don't like their bees to swarm and do all sorts of tricks to prevent that they will do so. And then we have this low breeding success of Varroa at high humidity. And so this is already good for the bees, and it gives a lot of possibilities for natural selection and for genetic changes in those bees that make that in the end they will become resistant against Varroa. There is something else which is interesting. That is that you have a lot of exotic bees in this country. And in our country we have also a lot of exotic bees. And that's because a lot of beekeepers for some reason think that those bees are better than our native bees. This is nonsense, but it is fake news spread by Brother Adam in the past, and it's still repeated. Very interesting, Brother Adam was active in the first half, mainly active in the first half of the 20th century. The same period was also used to develop the theoretical basis under evolutionary biology. But Brother Adam was an autodidact. He'd never studied, and he didn't know all these geniuses that lived here in England that were working on the theory. And those people, they had looked at whether there are genetic constraints on selection. And such constraints exist when you have very small populations. But if you have a population which is larger than, say, a couple of hundred breeding females, then there is no constraint. There is enough genetic variation in that population to select anything you might like. Meaning that you could select black bees that have all the properties that beekeepers that keep exotic bees think are only present in exotic bees. Exotic bees and black bees have the same genes. They have different frequencies of alleles, and sometimes it may take a little bit longer to select the properties in black bees, but it's possible. One of the consequences of the fact that black bees became very unpopular is that people did most of the selection, artificial selection work on buckfast bees and cardica bees and other exotic bees. And that means that the black bees were left alone. When I started to do something with bees, I was in a project that was, there is a reserve of black bees in the south of Belgium, and those people wanted to be part of that project. They wanted to do artificial selection. The first summer, we collected small bee colonies from all the different beekeepers, and then we studied if they had some resistance already or not. And the results are here. And the darkest things, you see there are colonies that were for 100%, they had 100% presence of Varroa specific hygiene. So you see that a lot of bees score already very high before any selection. A lot of bees also score 0. These bees come from different beekeepers, and some of them had already done a lot of selection work on their hives. But it shows that the black bee, because it has suffered less artificial selection, is a reservoir for resistant genes against illnesses. Here you see the distribution area of the black bee. It is an enormous population that covers very different climatic region. There must be a gold mine of genetic variability still present in our black bee. So now we are going to round up. So natural selection is possible if free mating large populations. That's the main message of this story. And in closed or sheltered populations, it works well, it works good enough, if only the population is large enough. And as we've seen in South Africa and in South America, wild honeybee colonies are very important for the evolution of resistance, because they are a source for natural selection. And as we said, the black bee is a reservoir of resistant genes.
I'm just interested to know what proportion of the audience here is also treatment-free, because we don't seem to have any Varroa in treatment-free hives in my area. There's quite a lot of people practicing treatment-free, and it just seemed to be like pushing an open door. It just worked.
I think we better point out that how long do you mean treatment-free? And don't forget, people bring in swarms and colonies and all sorts of things. So would you like to expand a little bit on what you mean?
12 years for me, a bit longer for other people. I'm in Oxfordshire, and we preferentially gather swarms from completely wild colonies or from our own hives. Then we swap them between the group to try and maintain a bit of, to reduce inbreeding. But they're still local bees, probably move them at most 10 miles.
Slightly more.
10% or so, yeah.
Okay, and how many people have not had to treat this year? That's more like 15 to 20, is it? Right, yeah. Out of about 100 people here. Thank you.
You quoted Derek Mitchell's research on the reproduction of Varroa in natural trees as being far, far lower. If I remember, the figure was on the screen at 2, if I remember. Therefore, I wonder, take the Arnott colony, Arnott forest example, and other wild colonies, if they're not so much Varroa resistant, but merely Varroa tolerant, because by the very nature they have far, far lower Varroa thresholds in the colony, and therefore they're tolerant to Varroa rather than being resistant to it, which I think is a big difference.
Yeah, that is. And I think that it starts out by being Varroa tolerant, but that makes that the colonies can survive, and that natural selection can do its work, and then in the end they are Varroa resistant. And we know that that happens because in retrospect we have measured now whether such colonies show a high frequency of the hygienic behavior and the allo and auto grooming behavior. And in all cases where I said they are fully resistant, those behaviors are very common. But the experiment that I mentioned that was done in Gotland, those bees, the surviving bees, they were not resistant, but they were tolerant. And they are tolerant in Sweden, and as soon as you bring them somewhere else, then they die. And that has to do with the fact that in Sweden the season is rather short, so Varroa has no time to kill the colony.
I'm speaking as a long time ago virologist, and I'm very interested to know what you think about the strains of deformed wing virus, because I know that some of the colonies and areas that are now resistant or tolerant are also those that do not have the virulent strain of the virus. Just wondered if you have any comments on that, please.
The work that was done in the UK showed that deformed wing virus type A has been replaced by deformed wing virus type B. Yeah. And a similar trend is happening in the USA as well. Yeah. Is it more or less virulent? No one's quite sure. And there's a type C, which is very, very rarely seen anymore. So that's it. So it's only A versus B that we're talking about.
But theory predicts that
sorry, so
a new virus.
So is there an impact? And the answer is no. Okay, so
yeah.
So we've now got type B in this country mainly, but we've now got a lot more resistant colonies as well.
But there is also the fact that viruses evolve to be less virulent over time, and because it's not in the interest of the virus, of course, to kill its host. So you would expect that in the long term, the deformed wing virus would be less harmful to the bees. But for that we need also natural selection.
Coming back to the point about relative humidity, you showed at 34 centigrade you've got much higher lethality for the mites at higher humidity. But there's a lot of work being done in Austria, and some work in this country too, about not just the humidity, but the temperature. Varroa mites are much more thermally sensitive.
Yeah.
So at 39, 40 centigrade, they don't survive for a 2-hour period. And if you raise the colony temperature, and we're talking about the brood temperature, above 40 for 2 hours, it doesn't affect reproduction of the bees, nor their performance.
But you should not do it too long, though.
For 2 hours, it's 2 hours. It's very easily done, 2 hours. But that would also correlate with your observations about the higher humidity.
Yeah, I know.
The thermal mass transfer from.
I know that some Greek beekeepers, they put their hives on a hot place in the sun for a while, just to kill the Varroas.
I'm a treatment-free beekeeper. I have been for 12 years now. I test for Varroa regularly. I run heavily insulated polystyrene hives, and I've measured for temperature and humidity. The humidity has always been up in the high 70s. Hive temperature, generally speaking, even in midwinter, is over 30, in summer 34. I find that I have a very, very low Varroa count most of the time. The odd spike, but you sit with it and it goes down. I use sugar rolls to test on a very regular basis. The question I've got is that are we doing our bees a disservice by keeping them in cold timber hives, and should we be doing a little bit more with insulation to keep the temperature and the humidity up in the hives, and as Derek Mitchell has often said, reduce the actual ability of the Varroa mite to reproduce? Should we all be looking at some more insulation on the top of our beehives?
Yeah, there are many reasons to improve the design of beehives. And this is one of them, indeed. We should. Beehives are designed for beekeepers to be able to work easily, but the design is not very appropriate for the bees themselves. I mean, they can live with it, but they can live much better if we adjust the design of the hives. And this afternoon I was discussing with Derek Mitchell that there is no such thing as a good comparative research on the standard of hives. We should do more so that we know what are the best hives, and that we should understand what is the best design.
I have to admit I was a little bit confused about this discussion on resistance versus tolerance. I think a tolerant bee survives without treatment against Varroa, and the same applies to a resistant bee. So what is the difference, and could you maybe give me the definition of, or your definition of resistance and tolerance?
I can do an attempt. The bees in South Africa, when the Varroa mite arrived, 50% or 60% of the colony survived. However, they had thousands of mites inside. So that must have cost the bees a lot in terms of energy. And now they are resistant. They're no longer tolerant because they are able to suppress the mite population and keep the mite population at a low level. So I would say somebody who is resistant, he can take care of pathogens and make that their population becomes insignificant. And tolerant would mean that you don't die, but you suffer.